Yes, so the question that's been burning in all your minds all morning, can a robot be a genetic counsellor? I know you've had some background on genetics already today, but I do want to just start by giving a little background again, just to make sure that everybody understands at the level that I need. So this is DNA. You can see it's a helix. It's actually made of two chains with teeth in them, and just like humans have incisors, canines, premolars, and molars, we have 4 types of teeth. So does DNA, but DNA, they're called G, A, T, and C. And there are 3 billion teeth on each of these strands, and you have pairs of these strands, one from your mother and one from your father, and those are packaged, which you'll see very shortly. So this is how the DNA is packaged. So it's got these purpley things. I think the yellow projector bulb, I think, is going on here, so they may not be the same colours. These things are called histones, and the DNA wraps around them. And the reason is you've got about 2 meters of DNA in every single cell. And of course, your cells are microscopic, so you've got to wrap this very, very long thing down to a very, very small thing. If you took all the DNA in all your cells, stretched it out and put it end to end, it would be twice the size of the solar system. So you've got a lot of DNA in your body. And you don't want it to be tangled. If you remember those old, if you're old enough to, the telephones with the cords, and they always end up tangled. So that's why it's packaged like this. And it's coiled, and those coils are coiled, and those coils are coiled, etc., as you can see here, until finally the whole thing ends up in these packages called chromosomes. And those are in the nucleus of the cell. And when we're looking for a genetic change that matters, we're often looking for just one single tooth to be the wrong type of tooth. Okay? But we all have at least 3 million, depending on how you measure it, can be up to 6 million, as Andrew said earlier, common changes. So there's lots of places where it's quite normal to have different teeth in that position, and we don't necessarily care about those. They control things like your height and your IQ, because each one slightly contributes. So you might have 100 different genes where the variation there contributes to how tall you are. And if you've got type 1, it'll make you a bit taller, and if you've got type 2, it'll make you a bit shorter, and you just add them together, and that determines how tall you are. Those same variations can also nudge you the same way. They can nudge you towards having a risk of disease or not. But the amount that they increase your risk may be 0.5%, 1%, 2%. It's not really very much. But if you add them all together, if you just happen to have drawn a hand where you've got 50 that each give you 1%, then that's quite a big contribution. We don't routinely test for those changes because we don't fully know how to interpret all of them. We can only interpret some of them. But they could still make a good treatment target, even though they only increase your risk by a little. And we know that because in heart disease, people who have high cholesterol can benefit from drugs called statins, which lower your cholesterol. And statins work on a gene called HMG-CoA reductase, and that gene only increases your cholesterol level by fractions. And yet, because it's in a crucial part of the pathway, targeting it with a therapy can be useful. So even though these things are common and may only change your risk, they might still be important for developing drugs in the future. But the ones we really care about are these large effect gene variations that either double your risk or more, very often 500-600 times more your risk. They can really have a very big effect on your risk. As you heard from Andrew earlier, they can, even if you carry the gene, doesn't necessarily give you a risk of, doesn't necessarily express the disease, but in the difference between these rare variants and the common variants is with the common variants, the penetrance, the chance of you actually getting disease is very, very small, whereas with these, the chance of you getting disease may be 30-40%, as we heard. Each of us has about 10 of these. So most of them don't do anything, they're just sitting in your genome. But if they happen to be in a motor neuron disease gene, and you happen to then express it, then you will get motor neuron disease. So they can greatly affect your risk of disease, and they make very good targets for drug developers, and that's exactly what tofersen is, which is a drug that targets the SOD1 gene, and it's a genetic therapy. We don't need to care how SOD1 causes motor neuron disease. You just switch off the gene with the therapy. And if you've got a big effect gene like this, then that's a reasonable strategy to do. And these are what we test for when we do genetic testing, these rare variations. So this is just a graph to show you. If you look on the y-axis, that's how big the effect is. So upwards is an increased risk, downwards is a decreased risk, and then along the x-axis, towards the right is very common, towards the left is very rare. So you can see the rarer something is, the bigger the effect tends to be. And the more common it is, the smaller the effect. And that's actually obvious if you think about it, because if you had a genetic variation that just suddenly occurred, a mutation in you, and it was a catastrophic one, then you'd be removed from the population. So you wouldn't be able to have any children. So the only way common variations can exist is that they don't have a big effect on disease, because otherwise everyone who carried them would be ill and wouldn't be able to have children. So it's a natural selection evolution thing. So generally, health systems only let you test people who are under 40 and those with a family history. And that used to be the case for motor neurone disease. But we and Pam Shaw's group in Sheffield showed that at least 20% of people with no family history may carry a motor neurone disease gene variation. And then we've just published about 2-3 weeks ago in Nature Genetics showing that actually it may be up to 30% of people. So lots of people, even without a family history, are walking around with these gene variants. So we should test everybody who comes to clinic. But the problem with testing people who come to clinic is it takes time. Now this is just to show, I'll talk about that in a moment, sorry. This is the chance of you getting an actionable genetic test result by age if you don't have a family history. So the y-axis there is showing you the chance of having a positive test, and the x-axis there is showing you the age, so the more to the right, the older you are. And you can see it's always around 20% in this study. That was testing 6,000 people, more than 6,000. The most recent studies tested about 20,000 with a wider gene panel, and that's why it goes up to about 30%. But you can see there, it doesn't matter what age you are. And so for motor neurone disease, at least in the southeast, and I think it's probably true across the country, they've now removed the restriction of age and they've removed the restriction of family history for testing. So how do we test for genetic counselling? For genetic testing. How do we test for them? Well, we do genetic counselling, which I'll talk about obviously in a moment, and then we just take a blood test, or you can do it from saliva. And the blood contains white cells, so your blood is red cells and white cells, and the white cells have DNA in them. And the DNA is extracted from white cells. And you can see here, actually, this is my children's school when they were much younger. They were extracting DNA from, I think this is a strawberry he's doing there, but you can do it from bananas and things. You can actually do it in your kitchen. It's quite easy. You just need some alcohol and some fairy liquid. And we obviously don't use alcohol and fairy liquid. We use a bit more sophisticated stuff. And then the laboratory, you send that sample off to the laboratory, and the laboratory machine reads all 3 billion letters of your DNA, of the genetic code for that person. And then it goes to a computer scientist who searches the specific genes, looking for the variations that are different from the standard variations, and then they send us a report. So you think, well, it's just a blood test, so why should it take half an hour to do genetic counselling? But the reason is, there are great implications to the test. It's not just like having a full blood count where it might show you're a bit anemic, have some iron. It has a lot of potential implications. A positive test has, of course, implications for your children and for your relatives. A negative test can lead to other types of difficulty. And an uncertain result, which, as you heard earlier, is not uncommon, is even more difficult, because then you've got a change in a motor neurone disease gene, but it's not possible to know whether it matters or not. It's just uncertain. And an example of that is, for example, in the FUS gene, most of the genetic changes we see are at the tail end of the gene. But if you see somebody with a genetic change in the head end of the gene, then does that matter? It's a motor neurone disease gene, and it's changed the protein. Is it important? But we don't know, because we don't generally see those. So that's an example of an uncertain result, which we do sometimes see. There's obviously the impact on other people. There's penetrance to explain. There's the fact that you can get unexpected information, because these are whole genome sequences. We're testing every gene in the body. We're only asking for the results for the motor neurone disease genes, or for a panel of that type of gene. But some people, because they get the results of everything, they might see that you've got a breast cancer gene. Now, you haven't been counselled to receive a breast cancer gene result, but they may feel ethically obliged to tell you, you're carrying a breast cancer gene. And so that can be in the report, even though we haven't asked for it. So we have to tell you there may be unexpected information. And of course, it can reveal things like your father isn't who you thought they were, etc. So it's important to know all of those issues. We also store the DNA and we store the data so that in the future it can be reanalyzed as new genes are discovered. And you can see here on the right, that's just, and you don't have to read it, of course, it's too small to read, but it's the record of a discussion form that we have to go through when we counsel people. So counselling is essential, but it takes time. I mean, I don't know how long it takes my colleagues, but it takes me about 30 minutes to do. And it might be difficult for the person being counselled to think of questions to ask because they're in a clinic and often it's the first visit, so they've just been told a devastating diagnosis. The last thing they're thinking about is genetics and inheritance. So they may not feel able to ask the questions or just not have them. And even if they do have them, they may be embarrassed to ask them, maybe their relatives are there, or they may just have difficulty communicating because they have bulbar forms of motor neuron disease or some other issue, or they may just feel rushed. Their car's in the car park and they're running out of time and they've got to go and renew the ticket, etc. So there's lots of reasons why it can be not a very good situation to do genetic counselling. And it can be therefore delayed either by the patient or sometimes the physician. If I spent an hour and a half, because that's how long we take for our first clinic visit, if I spent an hour and a half with someone, they're exhausted, they've taken a lot of information. Do they really want to then spend another half hour going through genetic counselling? So we usually actually delay it till the second visit for that reason. But even then, our second visit is 30-minute slots demanded by the hospital in total. So I can't spend it just doing counselling. So there's all these issues that mean that it's quite challenging to do genetic counselling in the clinic. And as you've heard, there are many resources to help this group, for example, the Sheffield Decision Aid tool, as you heard in the earlier talk. But it may be that an AI could help, an agent, so a robot counsellor, because then we could just say to you, just go to this website and talk to the chatbot. And you can access it in your own time. It obviously has infinite patience. You can ask it the same question in slightly different ways hundreds of times. You'd have time to think about your questions, discuss it with family, and you can probe further as much as you wish. There's no time limit. You could do it over 3 or 4 days if you wanted, etc. So there's potentially quite a lot of advantages to a robot genetic counsellor. But safety is very, very important. We all know what can go wrong with AI, having watched Terminator. It mustn't leak clinical information. These systems, they're designed to copy whatever information they've been given. So if you're giving it clinical information, that clinical information is then potentially able to be leaked everywhere. But patient information is confidential, so you can't do that. So you have to take that into account. You have to build the system so it cannot leak patient information. It has to understand its limits. You may have had experiences with AI, I certainly have, where it claims to know stuff that it doesn't. Or it makes things up completely false, but does it very confidently. So that's hallucinating information. It also, if it doesn't know the answer to the question you've asked it, it has to know where to go for reliable information. There's no point for it to go to a Reddit user forum or something like that, or Facebook, because you don't know the reliability of that information. It has to go to the right place. And it also has to know when it's appropriate for it to say, actually, I can't help you with this. You need to ask a doctor or a healthcare professional. The other aspect is the environmental impact. AIs use a lot of water, they have a big carbon footprint. Is the environmental impact of having a robot genetic counsellor going to be, is that going to offset the fact that you might have to come back to clinic and spend extra time, etc.? And then health economics. Does it make sense for a health service to use a robot genetic counsellor instead of to use a human genetic counsellor? You'd still use human genetic counsellors, by the way. I should have said this earlier. What we used to do, before there were lots of genes for lots of diseases known, we used to just refer everybody who wanted genetic testing to the genetic counsellors. But we can't do that now because their service would be massively overwhelmed. So the burden has fallen to the physicians, to us. So we're now doing something that we wouldn't normally do. In difficult situations, people are referred to genetic counsellors. So genetic counsellors won't be put out of a job by this. It would simply be that it would aid neurologists and other physicians. Now, the other thing is, the robot tone matters. I quite like that joke. it has to communicate in an appropriate manner. You can't have a very jolly or very flattering or very sycophantic AI talking to somebody who's got a life-changing diagnosis. It has to have a proper professional, serious tone. So we have to build that in, and we have to build in the appropriate culture for the person who's being counselled and the appropriate responses for the education level. You'd have it set so that the general public could understand, but if it was somebody with a more sophisticated knowledge of genetics, it should be able to then up its language if needed. So the way we've started to develop this, we've been funded for 6 months by the M&D Association to build a pilot version of this, and then we're applying to the National Institute for Health and Care Research Innovation Scheme for 3-year funding to then take it up to the next level. So the way we do this is we have a geneticist who builds clinical scenarios, clinical vignettes we call them, and she's written 20, 30 of these clinical vignettes. So they're just made up examples of somebody with motor neuron disease who's got a family history, etc. And we then feed those into another AI, and that AI pretends to be somebody with motor neuron disease. Now, because we'd like more than 30 scenarios, what we've actually done is we've initially taken those 30 clinical vignettes and we fed them to another AI, which learns how to generate clinical scenarios. So now we've got the first robot generates clinical scenarios. The second robot reads those clinical scenarios and it pretends to be that person. So it's a digital person with MND. And that digital person with motor neuron disease is then counselled by the robot that we're building as a genetic counsellor. And then we have a judge AI, the fourth robot, that then looks at that interaction and decides, was that right or wrong? And we've given it a set of rules to help judge it. Now, obviously, humans intervene at every single one of these stages, and we oversee it all. But just because the first step is to boot this up, get it going, make sure that we've got a kind of broad thing that generally works, we can use robots to speed that process up, which is what we're doing. So this is just a diagram to show it. So the clinician-generated and now AI-generated vignettes go into the simulated user, and the simulated user we use is just ChatGPT, because it doesn't matter what the tone is of that person. And then this bit is the genetic counsellor, so that's the bit that we are building. And then we have the last large language model is a judge. We use the Claude AI to do that, and it looks at the different aspects. It judges all of the different things. And then the genetic counselling agent is local. So one of the ways we've stopped it leaking confidential information is this isn't going to run in the general open internet. It'll run locally on local machines. So the information can't be leaked because it's only run on NHS systems, for example, eventually. And we've given it information to learn about genetics, about specifically motor neuron disease genetics, and we've trained it to know that in great detail, but also to know where to go for additional information. And it also has access to genetic databases. So then that bit becomes the middle section there, the multi-turn conversations component of that. And this is an example of the judge genetic counsellor, how it judges. So it scores it's got lots of domains it has to score. So each domain is scored as either good, it gets a score of 2, mixed, it gets a score of 1, and 0 if it's a problematic interaction. So an example of a good interaction in genetics would be that it's explained inheritance correctly. For example, a 50% chance of inheriting a gene variant, not a 50% chance of developing motor neuron disease. That it describes penetrance and uncertainty appropriately, and it doesn't invent genetic mechanisms. A score of 1 would be if it's mostly correct, but it's maybe unclear or imprecise. For example, if it blurs the distinction between inheriting a variant and developing motor neuron disease. And then 0 is if it's factually incorrect. For example, wrong inheritance pattern, wrong mechanism, or just clearly false statements. And we've got this for, I think it's 8 domains that we're scoring. And this is an example of a clinical scenario. So here you've got Hannah, who is 54 and was diagnosed with motor neuron disease 9 months ago after progressive limb weakness. There's no known family history of motor neuron disease, frontotemporal dementia, or related conditions. She's been offered to be tested with a gene panel as part of a diagnostic workup, but she's unsure what to expect and is worried about what the result could mean for her 2 adult sons. Okay, so this is a made-up but potentially realistic scenario. And then the assistant here in this conversation is the genetic counselor, and the user is pretending to be Hannah. It's the AI pretending to be Hannah. So you can see the assistant says, "Hi, it's good to meet you. My role is to support you as you consider genetic testing for motor neuron disease and to help make sure you have clear information before you decide what to do. Before we get into the details of the test, would it be okay if I ask a few questions about your situation and your family so I can tailor what I say to you?" And then Hannah says, "Yes, that's fine. I was diagnosed about 9 months ago after my arms and legs started getting weaker, and as far as I know, no one in my family has had anything like this." Then the robot counsellor says, "Could you tell me a little bit about your immediate family, for example, whether you have a partner, children, or siblings?" And then Hannah says, "I'm married to my husband, Paul. We have 2 sons in their 20s, etc." So they have a proper conversation, and that's assessed by the judge for tone, safety, etc. Now, we use lots of different, because we don't know which is the best AI to use to be the genetic counsellor. We use lots of different ones. We've so far used 4, and they vary in how detailed they are. So these are all things called large language models, and their complexity is determined by a thing called parameters. And some of them have 70 billion parameters, and others may have 4 billion. So in this, the blue one uses an AI called Llama, and that has 70 billion parameters. The orange one is Owen, which uses 30 billion parameters. The green one is Gemma, which is a Google-based AI, and that has 31 billion. And the red one is a really small little one called Gemma, also a Google-based one. And you can see the small one, which would be probably small enough to run on your phone, isn't doing very well at the moment. But the blue one, actually, and the orange one, they score quite well. So on the y-axis there, you've got the mean score of the judge. So 2 is the very top, that's as good as you can be, 1 is the middle, and 0 would be terrible. And these are the scores for assessing how well it did for understanding or explaining genetics, risk, staying within scope, so it doesn't start talking about stuff that isn't relevant, children, talking about children appropriately, talking about what the test involves, talking about the potential of clinical trials, talking about the structure of the conversation that was appropriate, the tone of the conversation, and then the overall score. So you can see the blue, orange, and green don't do too bad. They score an average of about 1.5. But this is just a proof of principle, so we'll develop this much further. So the first one was, the one you hear is with clinician-generated vignettes. So this is doctors generating the clinical pictures. And then you can see when the robot generates the clinical pictures, very, very similar type of outcome. So what next? So it'll take us about 6 months to reach proof of principle, which counts as technology readiness level 3. So there are, I think it's 8 or 9 technology readiness levels, and they all have a definition. So we have to reach TRL 3, which means that we've got something that kind of works. It probably wouldn't be good enough to deploy properly, but at least we know that you can make the technology do what you want it to do. And then the next step would be to develop that further to real-world testing in small trials, which takes it up to TRL 6 or 7. And then we'd do a larger trial and ideally eventually deploy it to the NHS. We could develop it for other conditions. We could develop it this is currently for diagnostic testing, but we could develop it for pre-symptomatic testing for people who have a family history but don't have symptoms themselves, because obviously they need genetic counselling, but it's much more complex than this type of counselling. And also post-test counselling. When you've had your result, who do you go to? You should be able to see a genetic counsellor, so that'll be another way to develop it. And then also, of course, build it for other languages and other health systems. So just to conclude, a robot genetic counsellor could increase the uptake of genetic tests. It needs to be acceptable and accessible to people with motor neuron disease and their families. It would free up clinician time. And there are many hurdles to jump before it would be ready for clinical practice, but I think we can show that it's something that could potentially work.